Chromosome synapsis. Individual 1. Individual 1 has the following chromosomes: MLI, centromere JKHG, and GHI, centromere JKLM. In this scenario, the period represents the centromere. So here's a drawing of chromosome 1 and chromosome 2. We had to add an inversion loop into the chromosomes so that each chromosome pair would line up, meaning that the GHKJILM chromatids line up in the same place on each chromosome. So let's take a look at the chromatids that are being formed. So here are the 4 chromatids that we get from this cross. We have red being MLI, centromere JKHG, purple being MLI, centromere JKLM, green being GHI, centromere JKLM, and blue being GHKJ, centromere IHG. The red and the purple chromatids look very similar to the original parent, meaning there's no extra addition or deletion of any genetic material, making the red and purple viable gametes. However, the blue and the green are reversed, starting with G. The green has the L and the M chromatid repeated, and the blue has the G and the H chromatid repeated. This means that there is additional genetic material, making these non-viable gametes. On to individual 2. Individual 2 has the following chromosomes: MLI, centromere JKHG, and GHKJ, centromere ILM. Again, in this scenario, the period represents the centromere. So here's a drawing out of our chromosomes. Since these chromosomes are mirror images of each other, they're looking very similar. We know that there is going to be a crossing over event, which is notated by the X in the middle of the chromosomes between the J and the K. So let's look at the chromatids that are being formed from this event. We have red being MLI, centromere JKHG, the green being MLI, centromere JKHG, we have the purple being MLI, centromere JKHG, and the blue being MLI, centromere JKHG. Looking at all these gametes, we can tell that there is no inversions with this cross, and all the gametes are going to be identical. There's no additional genetic material, and there's no deletion of any genetic material. So all 4 of these gametes are viable. On to the fitness of the 2 individuals. Looking back at individual 1, we saw that there were only 2 viable gametes. And looking back on individual 2, we can see that all 4 of its gametes were viable, meaning that individual 2 has the higher fitness of the 2 individuals.